All right. Well, welcome everybody. Thanks a lot for joining on our 6th iteration of the CMFI mass spec seminar today. And yeah, it's my very great pleasure to welcome Dr. Tomáš Pluska from the ICBM in Prague, who some of you know as like one of the main developers of MZmine. Yeah, I'm certainly a fan of his work, so pretty glad that he's gonna speak about feature finding today here in this guest lecture. And just to give you a brief background about Tomáš, I think he is a very unique path. After studying computer science at the Charles University in Prague, he decided to turn more into the direction of biology and perhaps karate as well at the Okinawa Institute in Japan, where he did his PhD and apparently perfectionized his karate skills before he then moved on as a postdoctoral researcher to the Whitehead Institute at the MIT in Boston in the United States. And now since I think 2 years or 3 years, he is an independent group leader at ICBM in Prague, and is running both, I think, biology-focused, particular plant biology-focused molecular biology lab, but also drives mainly the software development for mass spectrometry data analysis software, and in particular MZmine. In addition to now also running his own dojo, I've heard. So yeah, without further ado, I think it's perfect that you give this lecture today about feature finding and maybe a little bit about the brand new MZmine 3. So yeah, welcome, and I'm very much looking forward to your talk.
Thanks so much, Daniel, for the introduction. And I guess I'll share my screen.
Let me just unshare mine really quick. Okay, here we go.
Okay, so you can see my slides. Good. So thanks, Daniel, for inviting me, and just put this aside. Just a tiny detail, it's IOCB Prague, okay? But that doesn't really matter. And so what I'm going to be talking about today is not really what my lab is working on, namely. I mean, we are mainly working on plants and combining genetics with metabolomics to study natural products and the biosynthetic potential of plants and so on. But Daniel kind of wanted me to cover this topic, which is sort of fundamental and important for mass spectrometry data processing, and which will set the ground for next presentations about data processing with MZmine that Robin will be showing in the next seminar, and then feature-based molecular networking and so on. Okay, so this is going to be maybe a little bit technical, but I think it's a very important area to understand for people who work with mass spectrometry. Okay, so in the beginning, I'm going to cover the very basics, and then I'll go to the basics, and then I'll talk a little bit about evaluation. Okay, so first the very basics. So this is how LC-MS data looks like. I'll try to put a pointer here. Okay, so this is how LC-MS data looks like. Probably most people in the audience already have seen some data and are a bit familiar. Here we have the retention time. Here we have the mass, or the M over Z. If we slice the data in this way across the green line, it's called a chromatogram. If we slice the data across the red line, it's called a mass spectrum. And then we have some peaks, or I would like to say we have features here that correspond to some molecules. What I want to highlight here is that these peaks actually are 3D objects. So the data that's coming from the mass spec is continuous in both directions, in the retention time direction and the mass direction. So the features that come up are actually 3D, and you can see that data coming from different instruments have their own specifics. So in the Orbitrap data here, we have a better resolution, and then in this water synapse data here, the synapse data, on the other hand, is much more dense because the scanning is much faster than in the Orbitrap. And in the orbiter, we can also see some residual signals here that are called the shoulder peaks, that are the products of the Fourier transform algorithm in the instrument. Okay, so depending on the instrument, data looks a little bit different. Now, the goal of feature detection is to detect all these features. Unfortunately, the terminology in feature detection is extremely confusing. So in the literature, you will find any combination of these terms. And Daniel, as you have seen, Daniel's favorite combination is feature finding. I personally prefer feature detection. But the point here is that it's a little bit confusing. It is actually so confusing that there is a paper published just about how confusing the terminology is. So in this paper, they show that there are so many different things that you can call a peak in mass spectrometry or a feature according to the official control vocabulary. Okay, so this is bad. In this paper, what they propose as a solution is to design a completely new terminology. So they propose new terms like a neutromer, chargeite, and molecular Hall trace, and so on, which is an interesting idea, a terrible idea in my view, because then some people might actually adopt this terminology and then there's even more confusion in the field. Okay? So please don't do that, but this is just to highlight that the terminology is a little bit confusing. So to make a long story short, what I would like to call peak detection is when we are detecting signals in a 2D space. So that means we have, for example, time and some intensity, we are detecting peaks. If we're talking about 3D space where we have the additional dimension, like retention time, mass, and intensity, I would like to call that a feature detection, and the things that we are detecting are features, just to make things clear. And so the goal of the feature detection is to take the raw data like this that comes from the instrument and translate it into a table that can then be interpreted by a biologist or by someone who is trying to understand what's going on. So a table where we would have the masses, the retention times, and some sort of quantitative output like peak areas in different samples. Now, in order to do that, you definitely need to know a little bit more about your data. So you need to know what format is your data. We have different proprietary formats that come from the instrument vendors. We have some open formats like MZmine, it's maybe the most popular one, but also others. And the data can be in different flavors. It can be profile on centroid spectra, the mass resolution and mass accuracy can be different, and also the chromatographic resolution. Obviously, HPLC versus UPLC will be very different peak shapes. And then once you produce your feature list, you perform your feature detection, you might want to export your data somewhere. It could be an MGF file for uploading to GNPS. It could be MZtab file or feature XML, a format that's used by OpenMS. So there's it's good to have a little bit of background understanding of what is actually in data and what you're trying to do with it. So let's talk about it more in detail. I mentioned profile and centroid spectra. I suppose some people are already familiar, maybe some people might not be. So this is the difference between profile and centroided spectra. Here we are not looking at chromatograms. We are looking at actual individual mass spectra. So this dimension here is mass, or M/Z, against intensity. And the individual ions that the instrument is detecting come out as peaks. That's the continuous nature of the data that I mentioned. And this R value here represents the mass resolution of the instrument. Now, if we do a peak detection on the profile data and replace each of these peaks with a single line, it's called a centroid, we obtain a centroided mass spectrum. The advantage of this is that we reduce the file size of the data tenfold or even more. So it's a great way to save the space. But we also lose some information. So we lose the information about the mass resolution. And if the data is a bit noisy, we might lose some signals, like, for example, the tiny small isotopes might not be visible in the centroided spectra. Okay? And there is also a format that's somewhere in between where people don't do the full centroiding, but they simply remove data that's below a certain level, which they consider the level of noise. And then it becomes, as shown here, like thresholded spectrum, which still has the characteristics of the profile spectrum, but everything below certain level is missing, so there is no 0 data points and so on. This is an interesting idea, how to reduce the file size while preserving the information that's in the spectrum, but it's also quite difficult to process this data, because for the algorithms, it's hard to recognize this thresholded spectra. So I don't actually recommend anybody to do that. Okay, another thing I mentioned is the mass accuracy and precision. Imagine you do the measurement on a mass spec of the same ion again and again, so you get a distribution of mass values. And this here is the actual theoretical mass of that ion, and here is the average of the measurements that you did with this distribution. And so the difference is what tells you how accurate is your instrument. This is the mass error that you're detecting. And how wide is this distribution, that tells you the precision of your instrument. So how reproducible your mass measurements are. So to show this in a maybe more informative way, if you have a lot of measurements that are very tightly distributed and very close to the actual mass, the theoretical mass, then your instrument is accurate and precise. If you have still very close to the actual mass, the mean, the average value, then your instrument is accurate. But if your measurements are distributed widely, then it's not precise. If it's off, then it's obviously inaccurate, but it might still be precise if the measurements are very tightly distributed, or it can be inaccurate and imprecise if you have the value, the mean value is off, and also the measurements are not tightly distributed. And these are characteristics that you can observe from your own instrument. If you look at your own data, you see at the same ion across many different scans, see how much the values are changing. So you can actually learn this about your own instrument yourself. Okay. So this is the theory. And then why feature or peak detection is not straightforward. It sounds like a simple problem. I just identify those peaks in the data. But imagine you are the computer that's doing this, and then you get data like this. So in the first trace, we see a couple of dominant peaks with the same mass. So those are probably easy to detect. But as you start going lower, you start seeing some overlapping signals here. You start seeing signals at the very bottom where you're not completely sure if this is really a peak or if it's just noise, some random signal. And it gets more difficult when the data starts being noisy, like in this trace, going a bit up and down, not so sure how many peaks actually are there, or when we have a lot of peaks and a lot of noise with the same mass, or even here, is this really a peak, or is this just a bleed from the column, some sort of noise signal that appeared in the beginning and then disappeared? It's not easy for the algorithm to decide, because it's not even easy for a human to decide what is actually a good peak. So what are the challenges here? Now, metabolites are very chemically diverse. It's not like in proteomics where you're just dealing with amino acids, and you can even make some model, which in proteomics is called an averagine. So it's like an average amino acid. In metabolomics, we have such diverse chemical species that leads to also huge diversity in the data. So the peak shapes can be quite diverse even in one data set. The algorithms for peak detection or feature detection can have a lot of parameters. So with the same tool, different people can get different results depending on how they optimize the parameters. And depending on how you optimize the parameters, you're also tuning the sensitivity. So you can detect more features or less features. And that depends on how you set the parameters, but nobody can tell you what is a better result. Is it better to detect more features, even though some of those might be noise? Is it better to detect less features? Those might be reliable, but then you might be missing some features. It kind of depends on the problem you're studying. And then also when you start aligning samples with each other, you realize that feature detection might not be perfectly reproducible because you get features that you detect in only some samples, and then it's difficult to align them together. Okay? So I added this this nice meme that demonstrates how difficult it is to do feature detection. Okay, so to summarize the first part, feature detection is not a straightforward problem. It has a lot of underlying issues, and it helps you to understand the specifics of your data. So the mass accuracy, mass precision, mass resolution, what type of data you have, what data format, is it centroid or profile spectra, and so on. That's the practical part. So in the next section, I'm going to talk about the different algorithms that are used for feature detection. And I'm going to start with a bit of history. So I think one of the first published algorithms is MZmine from XCMS. This algorithm was mainly built for low resolution mass spectra, because at the time there were no high resolution instruments. And it was based on binning. So you take each spectrum and you bin it into bins of 0.1 m over z to essentially build chromatograms by binning. And sometimes it might happen that a single chromatogram is split into two bins that are adjacent, and that they try to address by combining the adjacent bins, as you can see here, to reconstruct such split chromatograms. Then when you have these chromatograms, you try to detect peaks in those chromatograms by using a matched filter. And matched filter is a technique from signal processing, which tries to correlate model signal, like here is this filter function. So it tries to correlate the model signal with your data. And so by doing this correlation, you identify that there is a signal here, and then that's how you identify the peak. Okay? This was one of the first algorithms, but not really suitable for high resolution data. Then XCMS got an upgrade with a wavelet transform approach. In case you're not familiar, probably everyone has heard about sine waves. So sine waves are just waves that go on forever. So there is an equivalent to that. It's called a wavelet. And wavelets can have different shapes. So this is the most widely used one, the so-called Mexican hat wavelet. And these wavelets are different from sine waves in the sense that they are localized in time. So this one goes on forever oscillating, but this one only goes once and then it disappears. Okay? And they can have different shapes, and the shape is defined by something that's called a scale in the wavelet. So with sine waves, when we have a signal that's measured, we can use an algorithm called Fourier transform to decompose that signal into a set of sine waves that together, when added, are going to reconstruct the signal. So in the wavelet transform, there is a similar approach. There is a similar approach that takes the signal and decomposes it into a set of wavelets at different scales and different positions that, when combined together, reconstruct the previous signal. And this is exactly what we need in feature detection, because those wavelets represent the features. And by adding them together, we reconstruct the original data set. So in XCMS, they first built chromatograms. They call them regions of interest by iterative methods. So whenever you see a data point, you're connecting the data points from the next scan and next scan and scan and so on. So you build these chromatograms, and then within those chromatograms, you apply this continuous wavelet transform to identify the waves across a range of different scales, because we know that the peaks can have different widths. And so this way, we find essentially the maximum of each wave across these scales, and that defines the position and the width of each peak. So this is a very classical approach to feature detection that has now been implemented in many different tools, but I think XCMS was one of the first ones. Now I'll describe something completely different. Okay, so Kalman filters are algorithms that are used, for example, in radars, where you have measurements of positions of aircrafts that are measured again and again, but every few moments. It's not measured continuously, it's measured with some period. And each of those measurements can have some uncertainty or some noise, but also you have to account for the movement of the object. So it's not only that you measure its position, but also next time you measure it's going to be somewhere else. So these algorithms are used to denoise and clear the noisy measurements from the radar, so you can actually estimate the position of the aircraft. Same thing you can find in self-driving cars nowadays. So there was this interesting tool that used the Kalman filter approach to create what they call pure ion chromatograms. That means chromatograms that belong to a single ion. And if you look at here, the mass spectrum, imagine that you detect an ion here. Okay? And same as in the aircraft, you're expecting the ion to move in the next scan. Okay? So you create a range of possible movements according to the precision of the instrument and the expected, peak shape going up or down. So in the next scan, you will be looking for that same ion within this area. And let's say you actually detected here. So you update your model and you say, I was expecting the ion here, I detected it here, so I'm gonna make some average around here. And that's how I update my model for the next scan. And then in the next scan, I'm gonna be expecting the same ion in here. And then you actually detect it somewhere here. So this is how you're tracking the ions across the scans using the Kalman filter, always updating your model according to the next measurement. So I think this is an interesting approach. It was originally implemented in MATLAB, but it was not open source, so it was actually not available for the community. And then in 2014, the Massive Quant project was published, where they actually implemented the same approach as a module for XCMS. So kind of interesting. On the other hand, when the same authors introduced a new version of their tool, TrackMass, they actually decided to abandon this Kalman filter approach, and instead they started building the chromatograms using the kind of iterative, greedy way similar to XCMS or MZmine. And for the peak detection in the chromatograms, they applied kind of a match filter approach that was, again, similar to XCMS. So it seems like this Kalman filter approach was interesting and sounds a bit cool, but maybe not necessarily the most robust way, because they abandoned it after some time. Now, another approach that is used is so-called adaptive signal processing. And that means you try to learn something about the data from the data itself. Yeah, so you look at the data set as a whole, and as shown here, you order all the MZ values from all the time points, and you plot the difference between the adjacent values, and you show the histogram. And then if these values are sort of randomly distributed, you would get a plot like this. But the actual data is going to show like this. And that means these values that are here are actually connected to each other. These belong to the same chromatogram. Okay. So that's how you can connect the points that belong to the same chromatogram and form the features from there. Seems like somebody talking in the background. Okay, another interesting approach that I thought was very different from the others was in this project called GridMass, where they took advantage of the continuous nature of the data as the 3D data set that I showed in the beginning. And here they do feature detection by placing a large number of probes. So these black dots are probes that are placed in the data set, and then they let those probes converge towards local maxima. And when many of these probes come to the same point where there's a local maximum, they say, okay, this area where those probes are coming to the same point defines the feature. And the same thing here. And so if your data is in the profile mode, is really continuous, you can use this approach to find the areas of the features without building any chromatograms and so on. So I think this approach is not super widely used, but it's kind of interesting. Now, another approach is based on k-means clustering. So imagine in your data, you have a signal intensity as shown here, and you select the highest ion in your data, and that's what they call the landmark ion. And then you look at all the other ions, and you plot their mass difference from that landmark ion. And you end up with a plot like this. And you can see in this plot, there are some clusters which are indicated by different colors. So those red dots here clearly indicate that one chromatogram where your ion belongs to, but there are a few other clusters. And so by using a clustering algorithm, you can separate these from each other and find even the chromatograms that are relatively similar in mass, but you can separate them from each other. This approach, I think, is actually used in several tools nowadays. And so as Daniel mentioned, I am heavily involved in the development of MZmine. So in MZmine, we took, from the beginning, we took like a 3-step approach to feature detection. So now I'm going to show you how it is in MZmine. So in the first step, we perform something we call mass detection, which is essentially centroiding. So you go to the menu, you select mass detection. There's a selection of algorithms that you can choose from. But for normal profile data, high resolution data, we typically use this exact mass algorithm. And here you can see your spectrum. And with these dots, it indicates where those ions or the centroids were detected. An additional thing that this algorithm can do, it removes everything below this noise level that you set here, but you can actually tell it to keep the fine isotopes that are detected even below the noise level, because those are useful later when you're doing feature annotation. So you can tell the software that for some chemical elements, you want to keep all the ions even below the noise level. Okay, so now you have all your ions in the spectra. And the second step is the ADAP chromatogram builder, which is a module that was originally developed by the Shusha lab at UNC Charlotte. And here you just need a couple of parameters. You're telling the module to connect all the chromatograms where at least 5 consecutive ion signals of the intensity above 1A3 appear after each other, and then we say this is a good chromatogram. And those have to be within this scan-to-scan mass difference. So basically 2 important parameters for this module. And the way it works is sort of inspired by the adaptive processing algorithms that I mentioned before. It sorts all the data points by intensity. It takes the next largest intensity data point from this list. And if its M over Z value is within that tolerance range of any of the chromatograms that we already constructed, then we add the point to the chromatogram. And if it's not, then we create a new chromatogram. Pretty simple and works quite well. And then after this step, you have built your chromatograms. And the last step is to identify the peaks within those chromatograms. For that, again, we have a range of algorithms, including some that are based on the wavelet transform from XCMS. But personally, the algorithm that I use the most is based on searching for local minima. So if you imagine this data shows the chromatogram, here we have a local minimum, and then here we have local minima, and here we have local minima, and so on. And so the points where you can detect local minima can be set as the points where you split the chromatogram into individual peaks. Here we have additional parameters, like for example, the peak has to have at least 3 times e⁵ signal intensity. So that's why this peak here is not detected, because it's below that threshold. And setting these parameters can be a bit tricky in the beginning, but MZmine has very nice help function where you can actually read about the meaning of these parameters and get more instructions about how to set them. Okay? And that's what you will see in the next seminar with Robin. One thing I want to highlight, I mentioned that the LC-MS data is 3D, but actually nowadays we're more and more seeing 4D data where we add an additional dimension of ion mobility separation. So ion mobility is a way how to separate ions according to their physical shape in a gas phase on a time scale of milliseconds. So it fits in between the separation by chromatography and then the measurement by the mass spec, which is in the scale of microseconds. So there are different ways how to do ion mobility. The most simple one is called a drift tube, where the ions are entering a tube where you have a gas that is slowing down the ions. Depending on the physical shape of the ions, it slows them in a different way. So in the end, they come out separated according to the size. Or you can have more sophisticated ion mobility chambers like the TIMS, trapped ion mobility spectrometry, where you have like more elements, let's say, and then changing voltages and interaction with the gas that leads to increased resolution in comparison to a simple drift tube. But the technology doesn't matter so much. Here we're talking about feature detection. So what I just want to highlight is that you can actually load this ion mobility separated data into MZmine. And you can visualize the data. You can see the separation both in the retention time and also in the ion mobility scale, and combine even multiple samples in this way. This is just a visualization tool. But you can also perform feature detection on these data sets. Now, the way how we do feature detection for ion mobility data is you start in a similar way to LC-MS data. You perform the centroiding or the mass detection. You get to your chromatograms by the ADAP chromatogram builder, and then you resolve your chromatograms into individual peaks. But then we look at the mobility dimension, and then for each peak, we expand the peak in the mobility dimension, as you can see here. And then we do another round of resolving. So the same algorithm that we can use for the chromatogram resolving, we can also apply in the mobility dimension. And we can, for example, find the local minima and split the features in the mobility dimension. And that's how we obtain then the individual features that can then further be aligned across samples. So this was mostly development by Stefan from the University of Münster, and it's actually one of the major features in MZmine 3. So if you perform this 4D feature detection in MZmine now, what you end up with is a feature table that gives you the shape of each peak in the chromatographic dimension, so across the retention time, but it also gives you the shape of the peak in the mobility dimension, as you can see here in the mobilograms. And you can display the traces across the retention time and the mobility and so on. So this is important because there are actually not many software tools nowadays that can do the feature detection for ion mobility datasets. So MZmine is one of the first ones. Another thing that we support in MZmine now is mass spec imaging. So you can actually load mass spec imaging dataset and you can display the images. And you can also perform a sort of feature detection on these images where it detects all the masses that are found in these images. And then for each mass, it creates an extracted image just for that mass. So then you end up with lots of images, which are not the typical features as I discussed before. They don't really have a retention time, but nevertheless, they can be considered something like features, those images. Okay, so this is the end of the second section. And what I would like you to take home from this is that there are many different algorithms for feature detection. And the most widely used algorithms tend to be based on a two-step process where you first build some sort of chromatograms, and then you detect peaks in those chromatograms. Seems like this approach is the most popular or most robust, let's say. In any case, however, it is worth spending some time to test these tools and algorithms and then decide what works best for you or for your instrument, because different instruments have different flavors of the data, as I discussed before. Okay, and in the last part, I would like to talk a little bit about how you can evaluate feature detection and how you can optimize it. So what you will encounter is that many of the methods that I presented require parameters, and those parameters have to be set by the user, and it's not always obvious how to set them in an optimal way. This is an example from the Centroid algorithm, from XCMS, or from the grid mass algorithm that was implemented in MZmine. So many different values that you need to provide. And why is it difficult to benchmark what values are optimal? And because you're not evaluating a single output, single metric, and you have to evaluate whether the features that you're detecting are quantitatively reasonable. That means they are increasing linearly with the concentration of the sample. And you have to evaluate the sensitivity and the specificity. That means whether you're not detecting too many features or too few features. And those two aspects have to be balanced. Also, we don't really have any data that could be considered a ground truth. So any data that you measure will always have a lot of noise, and there will be a lot more molecules than what you think there might be. And there is no such thing as a ground truth for feature detection. Some people tried to generate ground truth data sets, but for example, by simulating mass spec data or by doing a manual curation of mass spec data, but those approaches are also problematic, obviously. And if you do develop some way how to benchmark feature detection on your own instrument data, then you're obviously creating a bias for your instrument. It might not work same way for other instrument data. So what are the approaches that people have tried? So here in the XCMS paper, they tried a serial dilution. So they had a sample, and then they did serial dilution of that sample, which was some sort of extract from bacteria, I think. And they were measuring whether the response of the feature detection is correlating with the concentration of that extract. And also they were trying to use multiple tools or multiple algorithms, and then looking at the overlaps of the features, which I think is a good idea in general. I mean, trying different tools and then looking at what multiple tools can detect, and that's a reasonable way to go. So this is, I think, a nice way how to give a sensible outcome from your feature detection. It can be further improved, like as shown here in this paper, where they developed an automatized workflow based on design of experiment. So it's like a systematic way how to optimize parameters by repeating the same analysis with many different variations of the parameters, and then evaluating the results, in this case, the peak area versus some dilution or dilution factor, and then designing the parameters in a way that gives you the most reasonable result. So this has been done for XCMS. There is actually a tool for that, and also in this tool they tried a little bit different approach, looking at the natural isotopes. So every compound has carbon-13 isotopes, and you would expect that those isotopes would have a certain ratio from the main monoisotopic peak based on the chemical formula of the compound, and also the chromatographic shapes would be correlated. And so you can use these isotopes to benchmark whether your feature detection is going well, whether you're actually detecting all these isotopes. And using this tool, you can improve the parameters for XCMS to cover the complete features, let's say. So by doing the optimization, they show here that they could correctly split some peaks that were not split before. They could expand the shapes of the peaks that were incorrectly split before. So something like this using a design of experiment was also attempted for MZmine. But again, in this case, the authors decided to do a manual evaluation. So they tried to measure the number of peaks detected based on their own manual examination of the data set, which puts the human in the process, and that's actually very difficult and very time consuming. So I don't recommend this approach. Another approach that's pretty simple and that was used in the XCMS team is looking at the reproducibility. So you can probably expect, if you're analyzing the same types of samples, that your peaks would come up repeatedly because you're detecting the same compounds. So you look at how many features you're detecting and how many measurements, and probably you should expect a plot like this, where most of the features, I mean, if you have the same matrix, then you should expect actually more reproducibility, but they show that actually most of the features are detected only in a few samples, depending on your data set. I think for optimal feature detection, you should probably be trying to move this chart a bit more to the right, where you will be detecting the same compounds repeatedly. So it can be one of the matrix that you use. Now, obviously, the ground truth, if we have it, would be the best way how to evaluate whether the feature detection is working well. But some people try that. Some people try to actually examine a data set to a great level of detail. And here in this paper, you can see the authors spent several weeks of dedicated effort to annotate 2 data sets and list manually all the features that the human could detect. And then they evaluated the different feature detection algorithms, how many of those manually detected features they could find. Now, is this a good approach? I would say no. First of all, extremely time consuming. And also what they found is actually that MZmine was the worst performing feature detection algorithms according to their examination, which I find a bit strange because MZmine is used by thousands of people worldwide, and it definitely works if you use it well. So why spend so much effort on trying to optimize feature detection and then you get a result that's just not very reasonable? So don't do that. One more approach is based on examining peak shape. So you perform feature detection with different tools, and then you make a selection, like random selection of the peaks that you got, and you define some criteria. What is a good peak? So you define some objective criteria how to examine each peak, and then you look at how many peaks are actually good according to these criteria, and that gives you some output regarding, okay, ADAP versus XCMS versus the old MZmine 2 feature detection algorithms, how many peaks were actually produced that you can consider good. I think this has been done in quite a lot of detail, and was an interesting study that showed the strengths and the weaknesses of some of these algorithms. And finally, an interesting study that used over 1,000 pure standards that were spiked into a biological matrix. And here they evaluated a number of tools for both targeted and untargeted metabolomics. And they were looking at the overlapping markers that they could identify with the different tools. And here they concluded, in contrast with the other group that I showed previously, that MZmine outperformed all the other software tools in terms of the quantification accuracy, and they reported the most markers together with the fewest false markers. So interesting how different the conclusions can be. So the take-home message here is that evaluating feature detection is hard, and it's probably even more hard and more complex than the feature detection itself. And many people tend to rely on some sort of manual checking of the results. And I have seen this even in big labs where they do a lot of metabolomics. They actually have dedicated people to look at the feature detection results and then manually examine whether they are good or not. And I don't recommend to do that because it's, first of all, extremely time consuming and very subjective. Okay? So it's better to choose some automatic method. And what I would like to emphasize here is that the whole time today I talked just about feature detection. Okay? So how you detect those peaks, those features in your data. And I didn't talk at all about feature annotation, so I don't really care up to this point what those features represent, what kind of molecules. And so these topics will be covered in the upcoming seminars. So things like spectral library searching and library creation and molecular networking or feature-based molecular networking and ion identity molecular networking will be covered in the coming seminars by Robin and by Danielle. And so with this, I would like to thank you for your attention. This is just a picture from my lab with a lot of different plants. And I'll be happy to answer any questions.
Awesome. Thanks so much for giving this very comprehensive overlook or overview about the different algorithms for feature finding, or excuse me, feature detection. Yeah, I don't know, I'm sure people may have some questions. I'm gonna stop the recording in a second before we start with the question session. So this will not be on YouTube, but yeah, if you wanna ask, so please stay on. If not, I hope to see you all next time when, yeah, as Tomáš said, Robin is gonna speak about MZmine 3 and in particular how to do feature finding there in our hands-on session. So yeah, thanks again for joining and hope to see you next time.